Okay, so here is the first of today's lecture worksheets. So we're at 13.2 lecture. And let's break down some of what I've just covered for the structure of proteins. So proteins are just important, super important. They're not just in the food you eat, that you eat food and the food you eat contains carbohydrates, fats and oils, and proteins. The proteins are important in that you digest the food, and from the proteins get broken down not into the individual atoms. What proteins get digested into are those monomers, those amino acid monomers. And so not individual atoms, but the base unit, those monomers. So your body has then this repository of monomers that you've ingested, and then can use those monomers to build proteins in your body that are more useful for your body functions. So the human body can make most of those amino acid monomers. We talked about this in nutrition. So your body there are 20 amino acids and your body can make all but 8 of them. So those are called the essential amino acids. They need to be eaten, just like there are essential fats. So your body needs all 20 amino acids, can make 12 of them, but not 8 of them. And so therefore, there are some that need to come from food because food has amino acids, because food has protein. Now, not all foods have equal amounts of amino acids. So I talked about corn-based diets tend to not have enough of the complete or have the essential amino acids in them. So you have to have a balanced diet to get all the amino acids, particularly those 8 amino acids that your body needs. And most of us can do that through our basic diets. Okay, so this is the amino acid structure we talked about before, but now we're getting into the structure of proteins. Building the protein means putting amino acids together. So just like addition polymerization, when you bond one monomer over and over and over again, it's a little different with proteins because we have a name for the bond. So when you bond those amino acid monomers over and over, they're called peptide bonds. Okay, and the name of the polymer, instead of being called a polyamino acid, it's called a polypeptide. It's named after the bonds. Okay, because previously we've been naming, say, the synthetic polymers. The ethylene monomer becomes polyethylene polymer. Well, this becomes a polypeptide named after the bonds linking the monomers. Okay, so the peptide bond is what bonds, say, 2 amino acids. So here's an example of 2 amino acids. This is glycine and alanine. Okay, now for looking at the amino acid, you first want to remember that amino acid has a carbon with 4 different things linked to it. So this is the central carbon right here of this glycine in blue, and the 4 different things on the carbon of any amino acid monomer, 3 of them are always exactly the same. One is your NH2 group, so that's the NH2 group. The other is that's called the amine group. Okay, the other, this is also going off in blue towards the right, is your carboxylic acid group, that's COOH. And this, all right here, carboxylic acid. Okay, then you're always going to have a hydrogen and a side chain. So this is the side chain of glycine, is just 1 hydrogen. Okay, so the 1 hydrogen is always going to be on every central carbon of an amino acid. The side chain will be different, and this glycine happens to also be a hydrogen. Okay, so now the next amino acid that's going to be bonded is alanine, and alanine has this central carbon also. There's this on the left, the amine group, and then on the right is the carboxylic acid group. Here's the hydrogen side chain. So those are the same 3 for every amino acid. And then the side chain, what's different about the alanine amino acid is this CH3. So remember, that's what makes the 20 amino acids different, are the side chains. Okay, so they have different side chains. Okay, and they're not complicated. Like this one is just a hydrogen and this one is just a carbon with hydrogens. Okay, so what happens though in the formation of the peptide bond is that you have a covalent bond that forms. So that is where the carboxylic acid of one group, so here's the carboxylic acid of one of the first amino acid, is going to react with the amine group of the next amino acid and losing a water molecule in the process. So this will have to lose this OH group and at least one of these hydrogens, either this hydrogen or this upper hydrogen. So then what you have is hydrogen bonded to OH and the dipeptide. So let's draw that dipeptide where basically you have to cut off everything that's not the OH and the H that I circled in red. So let's leave everything else. So everything else is the hydrogen, the nitrogen with the two hydrogens, and then there's a carbon with two hydrogens, and then that carbon double bond oxygen. Okay, so here the red OH is now a bond. Actually, I'm going to make that red. This is a new bond. This is the covalent bond that's called the peptide bond that forms between the two amino acids. And so everything else of the alanine amino acid is still going to be the same. There's a nitrogen and now only one hydrogen. So there was a nitrogen and two hydrogens. Now there's only one because one hydrogen is in that H2O. And then everything else is the carbon with a hydrogen and a CH3. I flipped those up instead of down. That's because I ended up doing this zigzag up here. And then this is a carbon double bond oxygen OH. That's the carboxylic acid. So this is called a dipeptide because there's one peptide bond and there are two amino acids in this polymer. So there are two amino acids bonded together. So if I add a third one on, it's going to add on to one side of this polymer chain, and that will be a new peptide bond, and you'll have a tripeptide, and then you just keep on going. Okay, so as you bond these amino acids one at a time, this is a sequence of amino acids, and that is called the primary structure. So that's level one of the structure of a protein, is just taking the amino acids and sticking them together. Okay, so this original sequence of the amino acids, where did it come from? It came from the instruction that was coded on the DNA that was carried through on those triplet codons. Okay, so 3 bases mean put this glycine. A different 3 bases mean put this alanine. Okay, and so that's how you have the information of DNA. That secret code is read by building the amino acids one at a time on the protein. So this generic structure of this amino acid, let's just use the side chain R. So we're going to have to draw this primary structure now with 5 amino acids. Okay, so this, let's start with this on the side is NH2. I'm going to write it H2N like this. And so you have a carbon with a hydrogen, and then a side chain, and then this is a carboxylic acid like this. But now you just have the carbon double bond oxygen. Okay, so I'm going to actually put this peptide bond in red. And then this is going to the next amino acid, which is going to have its NH group. I'm going to put this here. And then it's bonded to the carbon that has a hydrogen and a side chain. And then it's carbon double bond oxygen from the carboxylic acid. But remember, you have to get rid of the OH. And there's going to be another peptide bond that's in red. Okay, so the peptide bonds are still in red. And then your next amino acid, which is the amine group minus the hydrogen. So that's NH. And then it's bonded to the central carbon that has the side chain and a hydrogen. And then it's what would be COOH. It would be the carboxylic acid, but because the OH group is gone, it's just carbon double bond oxygen. And then this is the peptide bond. And then the fourth one is going to be another bonded to the nitrogen, which still has one hydrogen. And then there's the central carbon with a hydrogen and a side chain R. And then another CO, that's the carbon double bond oxygen. And then finally another peptide bond. And this is to the last, did I count this right? 1, 2, 3, 4, 5, yes. This carbon is the central carbon with the side chain and the hydrogen. And now you've got COOH. So this is the carboxylic acid at the end on this side. As I mentioned in the video, there's a directionality of these amino acids, right to left, left to right, because it originated from a directionality, the reading of the DNA information. Okay, so that's the amine group on the left and the carboxylic acid group on the right, and this can just keep on going. You would bond the next amino acid to this carboxylic acid side. Okay, so that's the sequence of the amino acids. It's called the primary structure. So next, what happens is you have just basically a straight line of a bunch of amino acids bonded together. So that's your polymer. That's called your polypeptide. Okay, so that polypeptide is really basically just a straight, thick line with all these atoms bonded together. Well, next, what happens is a three-dimensional shape starts to form. So within that three-dimensional shape, there are repeated patterns called secondary structures. So secondary structure are repeated patterns that are common within a three-dimensional shape of a protein. An example is an alpha helix. An alpha helix is a coil. So that's alpha. We've seen that alpha before, but an alpha helix is a coil. And so that coil, we've seen it before in DNA, but DNA is a two-start coil and an alpha helix is just a single coil. Okay. Another example of a repeated pattern is called a beta pleated sheet. So this beta pleated sheet, that's the beta. We've seen that before too, with an alpha particle, beta particle, using the Greek symbols over. So the beta pleated sheet is like a accordion fold. So an accordion fold is basically, have you ever taken a piece of paper, here's my piece of paper, and folded it like this, like a fan? Okay, so I'm folding this like this, so showing this on here, and I've got this folded like this. So that's a beta pleated sheet in 3 dimensions. And this beta pleated sheet, drawing it on here, it would look like this. And it's not just going to be one of these sheets. It's going to have, another part of it. It could be the next part, but it has to actually be whole parts of the protein where these folds line up in 3 dimensions. And so these are repeated within a 3-dimensional structure of a protein. And these are basically common structures that you see in proteins, but not the entire protein structure. The way I think of it is like this is you have a house, right? And your house has a structure, it has a shape. It's probably different from the house next door. It's different than my house. It's different from the house across the street. And however, what everyone has in their houses is the same type of door. The door frame is always the same in most people's houses. Windows are generally the same in most people's houses. So those are repeated motifs found within the greater structures of general proteins. That's what secondary structures are. Okay, so you'll find an alpha helix in a part of a 3-dimensional protein structure. You'll find a beta pleated sheet in a 3-dimensional protein structure. The overall 3-dimensional protein structure is called tertiary structure. Okay, and so that's the overall 3-dimensional shape of one polypeptide. Just one polypeptide is one long sequence of amino acids. Well, then it becomes folded into a three-dimensional shape. Might have some alpha helix in there, might not. But that overall three-dimensional shape that looks like kind of a messy thing is not accidental. It is a deliberate three-dimensional shape. And it's held together by hydrogen bonds. And as in the video, you can have one polypeptide. Its three-dimensional shape is called tertiary structure. When you add in more than one polypeptide, let's say 4, which was the example of hemoglobin is basically myoglobin times 4. Then you are talking about more than one polypeptide's three-dimensional structure. Then you're talking about quaternary structure. And this is also held together by hydrogen bonds. So though it looks like kind of a big jumbled mess, it is not just randomness. It is held together by hydrogen bonds with this, amino acids. Side chains have the partial positive, partial negative, and those partial positive, partial negative attractions hold together the whole thing in exactly that three-dimensional shape, that structure. Okay, so this is amino acids that are probably very far apart in the overall sequence, because when you fold things in 3 dimensions, you get closeness of amino acids that were not necessarily close together in the sequence. Okay. All right. I think that gets me to everything. So the example of tertiary structure is one amino acid. Let's make sure you write that down. Structure is when you have the single structure of myoglobin. Okay, so it has its three-dimensional shape. Let's just draw it like that. Okay, the quaternary structure example is when you take that single amino acid, single polypeptide, and it has its three-dimensional shape, and you have more than one of those. Well, then you're talking about something like hemoglobin. So you take this shape, and I got to try to repeat this. Okay, and so I'm going to repeat this and have it in different colors. So these are 4 units compared to 1 unit. And it's 1 unit's three-dimensional shape, and then there's 4 units, that unit multiplied times 4, all of that three-dimensional shape together, and all those are held together by hydrogen bonds. And that hemoglobin is super important in all of us because it holds the oxygen, it carries oxygen in your bloodstream. Okay, so let's review all this on this genetic engineering worksheet for today. In this schematic, this is the process you saw in the video, but this is how human insulin was basically genetically engineered into this transgenic organism to basically grow human insulin protein that humans can't grow. So humans that suffer from diabetes don't make enough insulin, and they need that insulin. So how can they get that insulin? It needs to be grown. It can't just be something that's made in a lab or a factory. It actually has to be grown because proteins are complicated. So you need a living organism to grow that. So instead of just taking the insulin from a baby cow, you can train a bacteria, which is a living organism that people don't care if it gets killed off. You can train a bacteria to make a human protein. Now, when you have combined the genes from a bacteria with the genes from a human, that's why it's called transgenic. It's 2 different species genes. So it's nothing scary. It's nothing complicated. It's actually very easy because all of our genes, all of our DNA are made of the exact same nucleotides. Okay. So in this process, you have the chromosomes in a human cell are made out of the same DNA as the chromosomes in a bacteria. So those are 2 different species. Okay. And so You can take a human DNA with bacteria DNA. You can take human DNA with goat DNA. You can take daffodil DNA with bacterial DNA. You saw that in the GMOs extra credit. You can take corn DNA with bacteria, just 2 different species. Can be plant with animal, 2 different animals. Bacteria is single-celled organism, so that's not an animal or a plant. Okay, so the 2 different species here are human and bacteria. And bacteria is really the most commonly used because we can grow bacteria easily. And we can also grow bacteria easily because it is a single-celled organism. This is a single cell, and its DNA is on this circular structures called plasmids. So that's why it's called a plasmid, not a chromosome per se. Okay, so that's circular DNA. It's a circular DNA structure compared to an X-shaped DNA structure that we have on our chromosomes. So when you take out just the gene for making insulin, so insulin is a protein, and that insulin protein has a function. So it's, I think, classified as protein hormone function, because what a hormone does is it basically regulates something in the body. It can regulate the timing. That would be a steroid hormone, like a growth hormone is a timing hormone that is non-protein. But an insulin protein regulates the function in your body that allows for uptake of sugars. So you've got insulin protein that drives the function, that drives the timing that regulates another function in your body. Okay, so this insulin protein is coded by this gene. This is a stretch of DNA. It's anywhere from the hundreds to thousands of base pairs long, and this piece of DNA is the specific code for insulin. Okay, so then it is inserted into the plasmid, which is also DNA. Okay, so this is circular DNA, remember, and all of that works together because they're all the same parts. They're all the same nucleotides. So this is called recombinant at this point because it combines the DNA from one species, the human species, that's the yellow. So this is the human piece of DNA with the circular DNA from the bacteria. That's the blue. So now it's called recombinant DNA when you have the 2 different species. Okay. Now, when you take that recombinant DNA, which is mostly the circular DNA from the bacteria, you can put it back into a bacteria, and the bacteria doesn't know that that human piece of DNA is part of it. So the bacteria, this recombinant DNA is now inserted into a new bacteria. And this bacteria, just this volunteer bacteria, which you don't actually have to get to volunteer because it's just a single cell. So there's no ethics considered with this. This is going to live its life. What the bacteria does when it's living its life is it grows its proteins. How does it grow its proteins? Same as you and I. It expresses the DNA from DNA to proteins. And in the process of making proteins, it will end up also making this, following this instruction to make insulin protein. So this is making insulin protein is just a side product of this bacteria living its life and making all the regular proteins. Okay, so the label that sells that, let me make that grow the insulin, human insulin protein. So these are the cells. These cells that, these are the bacteria that have the recombinant DNA in there. Well, what bacteria does is it replicates, it makes 2 copies of itself, and then it goes on. And you don't actually have to insert that recombinant DNA into each individual bacteria. You insert it into one bacteria, it splits into 2 bacteria that then splits into another 2 that splits into another 2. And you just have to start with the one bacteria having the recombinant DNA inserted into it. Okay, so that all this bacteria that follows in their generations, the many generations. Okay. And the bacteria basically will replicate, it will duplicate itself every 20 minutes or so. And so however long you grow that bacteria culture, it can be for 20 minutes or for 36 hours, you will get lots and lots of bacterial cells. So from this bacterial growth, this bacterial cell growth, those bacterial cells are growing, they're doing their thing. And in those bacterial cells, now you have this insulin protein. So then what you do is you collect all those bacterial cells, you break them all apart, you open them all up, you get what you want, which is this insulin protein. You isolate that protein, and then you have that human insulin. That's what you wanted. Okay. Now the bacterial cells will end up, they live their lives. They naturally die off, and that there's no ethical considerations with that. Okay. So that's what happens with this, with genetic engineering, mostly done in bacterial cells. Pretty simple, pretty basic. And to make for this early was this making the human insulin, which was needed at the time and still needed. So it's called humulin, is the human insulin protein. Okay, so then what you do is you isolate humulin. That's the human insulin protein that comes out of the recombinant DNA that was grown in the bacteria. Okay, so let's pause. So yes, there's pros, there's cons. GMOs are with us, and exactly what are the good and bad. Okay, so first, genetic engineering. Is it natural? It is natural. Okay, so this genetic engineering has happened in nature. That there's sweet potatoes actually naturally there's an example of sweet potatoes that incorporated bacterial DNA into their sweet potato genome. So that's plant plus bacterial DNA. And this happened naturally 8,000 years ago. So let's not say it's not natural, okay? Also, the idea in general that we have been engineering plants for thousands of years, not transgenically, but by basically doing crosses of plants. I've talked about that at the outset, where we have corn, the corn we have today, that most of us eat corn, or we eat animals that have eaten corn, is because of the engineering of what had been a wild rice called teosinte. So that is thanks to engineering, but not in a lab, but by mixing DNA from different species to get the best possible crop, which is the corn we have now. Okay, so do GMOs cause health problems such as allergies and cancer? Though there is a theory that makes sense that GMOs would cause health problems, because your body is trained to recognize unknown or foreign antigens, which we'll get to next as bonus. There's so far no medical evidence of this. So yes, that's a theory. There are tight regulations on all lab created foods. And those are, let me add, they are FDA. So remember the good old FDA that has, mandated the nutrition facts label. Well, they also have tight regulations on all lab created foods. And that is including foods that are created at the DNA level when they are genetically modified. Okay, so this is a no. Okay, this is also a no. So do GMOs cause farmers to overuse pesticides and herbicides? The claim is that no, they don't. In fact, there's actually reduced reliance on these are chemicals, pesticides and herbicides, There's reduced reliance for those chemicals like Roundup, that's a pesticide, because of the use of the genetically modified Roundup Ready corn. Okay, and or using BT, genetically modified corn. So that's called BT corn. BT corn can make its own pesticide as the corn grows. And so when you eat BT corn, it doesn't affect you, that pesticide. It only affects the pest, the Bacillus thuringiensis, that corn, that European corn borer worm, but it doesn't affect humans. Okay. So it allows for not having to spray corn to kill off that worm, because that worm dies when it eats the corn that has been engineered for that pesticide just to kill the worm. So there's a really reduced reliance on chemicals, and that's a good thing. Having to spray less chemicals on farms, we like that. Okay. And it does not affect humans when they eat that BT corn. No medical evidence. Now, GMOs actually do create super insects and super weeds. And this is because species do co-evolve in nature. They are known to basically say, well, if I can't grow, if I can't survive eating this corn, then I'm just going to have to adapt. And so, yes, those are examples of super BT corn borer worms. And then also super weeds you saw in the video. So now this is also valid that farmers cannot replant those GMO seeds, that they, if Monsanto has made this BT corn, they have to buy those seeds every single year from Monsanto. And so that Monsanto, this company that makes BT corn, these seeds must be bought every year. I think what farmers plan on or hope to do, what they did in the past before BT corn, before genetic engineering, is they'd take some of their corn crop from the previous year and save it, because those corn become seeds for the next year. And so they can, instead of selling all their corn, they can save some of it and grow the corn crop for the next year. Well, this, the genetically modified corn, the BT corn, has to be bought every year. And so that becomes expensive. And so that becomes expensive for farmers, for farming in general, and that gets passed on to consumers. So that is a problem. And so in these debates, as with any debate, there's pros and there's cons. And the fact is that GMOs are common in the US. 92% of corn is genetically modified corn. 94% of soy is genetically modified soy. And you might say, well, I don't eat corn, I don't eat soy, but you actually do, because corn is used, remember, to make high fructose corn syrup. And so it's used in other foods. It's used in processed foods, HFCS. I showed you that in a video back in module 11, how high fructose corn syrup is in just about everything. It's in your salad dressing. It's in your soup. It's in your ketchup. And it is because it is used as a, to improve flavor and texture and shelf life. So that's coming from corn, and that's typically going to be those 92% of genetically modified corn. And then 94% of cotton, so that's cotton fabric, is genetically modified cotton. So, and that's separate from the organic, and that's hence the labeling that I mentioned in the video. And so labeling is controversial, because if there's no medical evidence proving that GMOs are dangerous, then why do we have to label? Because people are afraid of science in general, and they don't understand, and they need to be told, they want to be told what is potentially dangerous. And that's why GMO crops have been called frankenfoods. And I don't agree with it, but of course you guys get to make your own decision on that. And so that's the thing, is getting you to the point where, as a Chem 9 student, you get to make your decisions about nutrition, about the environment, about your energy sources. And, that is part of the sustainability part. But also, the idea of Chem9 is chemistry and understanding those chemical reactions behind climate change, behind ocean acidification, or even behind recycling. And so the basis of all this is understanding chemistry and chemical reactions. So I hope that when you see change, you'll link it to the idea that it's a chemical reaction. And so that's the idea, is chemical reactions are usually the changes you see. And what's going on is atoms are rearranging, bonds are broken, and new bonds are formed. And so that's just, I got two parts there that I hope when you are out there, you think about sustainability, but you also think about chemistry. And so in your daily choices, thinking about your choices with regards to climate change, air pollution, water pollution, and ocean acidification, where we can make those changes in our lives and make things better out there in the world. So, and this will also affect your immediate health, your nutrition, recycling, your electricity use. That'll affect your bills that you have to pay, and that's all good, right? Okay, so let me actually pause